So we have a few things to think about. And one is balanced cluster sizes, and another is highly similar clusters. Really think about the distribution we have in our data. Because if we take this example here and we do a differential expression, then cluster 1 and cluster 4 probably have a lot of the same genes coming up in both of them because they are a similar cell type or the subtypes of the same cell. Which also means that when you test, my point, cluster 4 versus 1, 2, 3, all the genes that are in cluster 4 might also be in cluster 1. So they will be less significant. So really think about how the relationship between your clusters is and what it means, what you get out. And if we go even further and have this, it might actually be the case that you don't get that many differentially expressed genes for cluster 4. Because when you take 4 versus rest, the rest is so dominated by all the cells in cluster 1 that it will be very few genes that come up for this cluster 4. And of course, there are ways to deal with this. And I would say it's probably a good idea to subsample the clusters. And actually in Seurat, there is a command to say max cells per ident. However, when it actually does the subsampling, it does it each time it runs a test. So we'll take still all of these cells here and subsample to say 50 cells or 100 cells, whatever we set. So it still will be dominated by cluster 1. So you would have to subsample first to have a fairly equal number of cells per cluster, and then run the find all clusters to have a more fair comparison. It's also the case that when we compare cluster 3 to rest, it will mainly just be what's differentially expressed between 3 and 1, and not so much what's different between 3 and 2. So think a bit about what dominates your data more, and think of strategies. Quite often also, if we have a case like this, you might not get that many or very similar genes for these. So it's more interesting to just do cluster 4 versus cluster 1, and not the 1 versus all, because these might get almost identical gene lists. So quite often it's a combination. I start with the 1 versus all, and then start digging into different populations and do a lot of additional differential expression tests between individual clusters and so on. More things. I said this about going back to the RNA-assay. In most cases, the normalized counts, log normalized. Read up on the methods if you're doing anything else than these standards in Seurat, which slot you should use. And as we talked about in the normalization exercise, what normalization we do does actually affect what results we get from differential expression, especially when we have very different cell sizes, very different big differences in the number of detected genes between two cell types. And with regards to batch effects, one thing is check that your genes are not just batch effect genes that are coming up if you're running a test. If you plot it not only per cluster, but also per sample, and you see it's upregulated in this cluster, but it's only upregulated in cells of this cluster that comes from one batch, then you really need to think about how to interpret it. And then also to use a test that can control for batch effects. So then not the Wilcoxon. And in Seurat, you have this latent vars parameter, where you can basically provide a vector with the names of the metadata you want to include, like the sample or the condition. And then also, if you want to, for instance, test for disease versus control or whatever within your clusters, then you would just subset for that cluster. So there's an example of that in the exercise. And then also, this is really going back to really plan in your experiment. How many cells do you really need to get good detection of differential expression? And that kind of depends on the level of expression also. Because for highly expressed genes, we have a larger chance of detecting them. There will be less of this stochastic transcription. So maybe it's enough with 20 cells in 2 clusters and comparing them to detect the difference in expression level. But when you start going down to the more lowly expressed genes, I would say probably 50 cells per cluster is required, if not more. And then, of course, what quality your data has, the higher quality, the bigger chance of detecting differential expression. And the more sensitive the method you're using, the better your chance of detecting something. But is it really that you want to look at differential expression between broad cell types, or is it very similar cell type? Then that also influences how many cells you need, because there might just be subtle differences between them. And also think about that in most cases, if you take 2 random sets of cells and run one of these tests, there will be a few genes that come up. So really think, is this just noise, or is it an actual biological signal? Does the genes make sense? So that's about differential expression, and then we'll continue a bit more on how to make sense of it. Do you have any questions for now?